Hey guys, hello today and welcome to our next histology practice. And so today we will be dealing with something that has a lot to do with the reproductive system that we did last time. So we will be moving on to the development of the fertilized egg, and we will today cover everything besides the embryo itself. So we will be talking about the so-called extra embryonic organs. So that means we will be talking about the umbilical cord that we have already seen in the practice dealing with connective tissue types. Then we will focus on the placenta and different stages of placenta development, and finally the fetal membranes. So the presentation that I will be using is uploaded to Výuka. I put it there yesterday evening, I think, so you should have it there as a PDF file. And if you will have any questions today, you are welcome to ask them in the chat that I will be checking from time to time, hopefully frequently enough. We will also be working as always with the workbook for summer semester, so you should have that on you as well. And I also recommend the PDF file that you also have on Výuka, which is labeled Extra Embryonic Organs Slides. So it's a PDF of labeled slides for today's practice. So I will be using some of those pictures today as well. So that should be it. The video will again be available when we are done recording this as well, so you don't have to worry about missing something. So the extra embryonal organs develop from the zygote. So when the oocyte that we talked about last time gets fertilized in the oviduct, the fallopian tube, it becomes a zygote, which will then start growing and dividing, and it gives rise to the embryo and the extra embryonal organs, and these start to develop during the second week of the development of the future embryo. They are not part of the embryo, but their function will be to nourish it, to support it, stuff like that, to connect it with the mother. And they grow very fast during the first period of the gestation. So you probably, I hope you went through the lectures as well. So we will cover some of the theory, but you have it more in depth in the presentations for the lectures. So we will be talking about different fetal membranes such as chorion, amnion, and allantois. We will also deal with the yolk sac and the umbilical cords, just some terms that we will be talking about. So we start with the oocyte, which came from the Graafian follicle by ovulation. Then it becomes the zygote. So zygote is the diploid, fused cell that is made from the oocyte and the sperm. That was the fastest one, best one to get in, and then it starts dividing. So it starts dividing, and after some time it will become this multicellular unit, which is called the blastocyst. So this is where we will start talking about the different parts of it and what they will give rise to. So the blastocyst is what we are interested in. And once again, reminding you about the location of all of these processes. So we come from the ovary into the infundibulum, this part of the oviduct, as the oocyte with zona pellucida, with the glycoproteins, and corona radiata, so the remaining follicular cells come from the Graafian follicle out into the oviduct. Fertilization happens, and then zygote forms and starts cleaving. So the cells start dividing. And it reaches the stage of blastocyst, and ideally we should be in the uterus now. So here we will be talking about what happens to the blastocyst and how does it integrate itself sort of with the lining of the uterus, which is the endometrium that we also talked about last time. So the process in which the blastocyst sits on the wall of the uterus and starts building itself into it is called nidation. And I have it in Czech here, sorry. So nidation is the process, and it comes from Latin or Greek, I think, and it means a nest. So it's sort of forming a nest inside the wall of the uterus, the endometrium, with the endometrial glands, the uterine glands that you can see here. I have a short video which I think might be good for the 3D orientation in this process. So it's like only 50 seconds. And so here we have the morula. So still a lot of cells and dividing even more. So now we shedded the zona pellucida, so the glycoproteins are gone, and the blastocyst is sitting onto the endometrium now. And here we start to distinguish between different areas or parts of this unit, this future embryo, sort of. But these are terms that we will be hearing from now on during the placenta and the embryonic. The fetal membranes, sorry, I'm confused. So we start seeing that there is a cavity and then two parts of cells which are called the hypoblast and epiblast. So based on their location, epi is usually something like more towards the surface. So epiblast is here, hypoblast closer to the center, and then we have cytotrophoblast and syncytiotrophoblast. So these two guys we will be meeting later on. So it starts sort of growing into the wall. So this is the nidation taking place. The whole multicellular blastocyst goes inside the wall of the uterus, and it sort of grows out into the tissue and starts forming what will become the fetal membranes and the placenta. So I will maybe add a link to the YouTube video as well, to the PDF, so that you can, it is useful anyway, watch it. So in the end, when it starts eating its way through into the endometrium, we will start seeing one more cavity here, and this will be the amniotic cavity. So amnion, chorion, allantois, the three layers of cells, and inside the amnion we will start seeing the cavity which will in the end become the cavity in which the embryo will be floating, sort of. So amniotic cavity and the developing proliferation of this into the maternal tissue. So this is still maternal tissue. This is the tissue of the embryo, the future embryo. And here we start seeing something which will be in future called villi, and we will have it in the placenta. So by the second week, we have something like this. So we have the blastocyst sitting in the endometrium, and there we start to divide the part which will become the embryo. So this is the epiblast and hypoblast that was even visible in the video. So epiblast and hypoblast will be part of the embryo, and trophoblast is the part which will give rise to everything else. So cytotrophoblast and syncytiotrophoblast. These will be forming placenta and stuff like that. So we are embedded in the endometrium, and what happens then is the whole structure starts growing. So here we had the second week, here we are progressing, and you see that this whole structure, so both the cytotrophoblast and syncytiotrophoblast and the embryoblast start growing, and the amniotic cavity starts growing as well. So this is what's happening before we have a look at the embryo or the extra embryonic organs on the slides. So this is just for your orientation, and you see here again that the tissue of the trophoblast starts growing into the endometrium, and it will be forming these villi, which we will meet in the placenta. So all of the green and pink and all of this is part of the blastocyst that developed into this huge structure, and the tissue around is still maternal. But the tissue of the mother is also changing. So the blastocyst came here to the epithelium of the endometrium, and it started a reaction of the endometrium itself, which is called the decidualization. So the endometrium will change to be able to provide nutrition and support for the developing embryo. And this is the decidualization, this process, or the decidual reaction. So following the nidation, so the landing of the blastocyst and it going through the surface into the uterine wall, the epithelium and all the cells of the endometrium will start to change. And what will happen? The cells will sort of swell. So here on the left you have stromal cells. So stromal cells in endometrium will be special type of fibroblasts, stuff like that. And when the nidation will happen, they will start storing a lot of glycogen. So they will fill with glycogen and lipids, and they will become from cells with this spindle-like shape, they will become round and sort of, so polyedric means they will not be perfectly round, but they will have many angles, but they will not be elongated like this. So decidual cells, so those will be the cells of the mother, of the endometrium, that we will see on the slides, and they will be large and swollen, so the cytoplasm will be very wide and pink. And the place where the blastocyst entered the endometrium is closed with fibrin, so clotted blood. And this place is called the operculum, but I don't think this is very crucial for us now. So you have to know what is decidualization or the decidual reaction, and so what happens to the cells and why. And so you can imagine, let me just check if there are some questions. Yes, nidation is the implantation. Yes. I think the implantation, I'm not 100% sure right now, but I think nidation is more of the going in, whereas implantation is just catching of the blastocyst on the surface and then it goes in. So we have a question about the nutrition for the not the egg, but the developing embryo. So the embryo doesn't destroy the cells, but the cells they store the nutrients and they can exocite them for the embryo to be able to utilize. Yeah, so I will repeat the decidualization once again. So I hope I answered the first two questions. So the decidualization is a process when the endometrium, when the endometrium registers, like there's a signal coming to the cells of the endometrium, which will tell them that a blastocyst, like fertilized egg, which has already divided many times, has landed on the endometrium, has arrived, and that the cells need to start changing to be able to make it stay, to make it survive. And this, what happens to the cells of the stroma. So the stroma is the non-functional part of the endometrium. So the cells of the stroma will start storing a lot of glycogen and lipids for the nutrition of the embryo. So they will swell because of this. They will just store so much glycogen and lipids that they will become big, and they will change shape and change size. So they will start looking different, and they will start to be called decidual cells. So this is the process of decidualization, is that we come from normal stroma to decidual cells that are ready to feed the embryo, the blastocyst that has nidated into the endometrium. So I hope that's clear. Yeah, so they just swell and store a lot of glycogen and lipids. And so based on which part of the uterus are we talking about, something a little bit different will happen to the decidua. So decidua is now the endometrium, which will be playing a role in providing nutrients to the embryo. So it will become part of placenta, and it will become part of the wall, like it will line the whole wall of the uterus and form the fetal membranes as well. So in the place where the blastocyst has nidated, so where this all started, the endometrium will be called decidua basalis. So it will be this area. So decidua basalis will be here at the place where the egg, the fertilized egg, first came to the endometrium and catched onto it. And this part will in the future become the maternal part of the placenta. So on this side, the placenta will start to develop, but all the rest of the wall of the uterus will also change. The change will happen to the cells in the endometrium everywhere. So endometrium is lining the wall of the uterus, and here it will be called decidua basalis, because it's the base of the place where the nidation happened. This will become part of placenta, and then the rest will be called either decidua capsularis or decidua parietalis. So you have already met the word parietal or parietalis before. So it usually means that something is lining the wall. So the wall of the whole uterus here, where it's not the future placenta, will be called decidua parietalis. So it's this thin layer of changed endometrium, which is now, when we have the developing embryo, called decidua parietalis. And you also, because if you remember from the video, the blastocyst didn't stay on the surface, it went underneath the surface of the endometrium. Therefore, you also will have a layer of endometrial cells that will change into decidual cells here. on the surface of the cavity where the embryo will be developing. So this part is also decidua because its maternal endometrium changed like this, and it's decidua capsularis. So it's covering the fetus. So this layer of endometrium on the surface of the fetus is decidua capsularis, so it's like a capsule around the embryo. Decidua basalis is on the base of this whole thing with the embryo inside, and it will become the placenta. And decidua parietalis is lining this free wall of the uterus. So this is kind of complicated for 3D imagination, but it's I think if you understand it, you don't have to memorize the words. It should really make sense, at least a little bit. So is it okay so far? Hopefully yes, we will see. So we have decidualized. The decidual reaction happened to the endometrium, and we now have 3 parts of the decidua where we have decidual cells, and it's basalis, parietalis, and capsularis. What we will see on the virtual slides in a while will be the decidual cells under the microscope. So if you remember from the last time, from the uterus slide, we just saw like something like this on the right between the uterine glands. So it's sort of loose connective tissue, lots of cells, blood vessels, nothing really out of the ordinary. And this part here is a part of the endometrium that's undergoing the decidualization process. So here you have a magnification of like area like this, an area like this. So this is still the stroma of the endometrium, and here you start seeing these huge cells. So they are not round but polyedric. You see some sort of angles, so that's like just to say that the term is right. It's not nonsense. And these are the nuclei stained with hematoxylin because they are acidic, and cytoplasm is very eosinophilic, very light, and it's light because of the lipids it's storing. They will not be stained, they will wash out during the preparation of the slides, if you remember, white adipose tissue. So here it's not just adipose tissue, it's not just lipids, it's also glycogen. So the cells are swollen like this, they are very big cytoplasm, and so they will be very easy to recognize. So whenever we meet something like this, either in the placenta or in the fetal membranes, this will be the cells of the decidua, of the maternal part of the tissue, because it's just a very changed endometrium. So it's still the maternal part. It's part of the wall of the uterus. So this is decidua under the microscope. No more questions. Perfect. Oh no. Sorry. So, if we move on, so what else do we have here developing from the blastocyst? So we already talked about the maternal part, the decidua, and then we have the fetal membranes. So one of them is the chorion. So chorion is here is the dark reddish color. So the orange is the maternal, the decidua, and now the biggest part here that will be forming the future placenta, and it's also surrounding the embryo, is chorion. And chorion has 2 sort of sides, so it has 2 parts. And on the inner side, it will have a tissue that's called extra-embryonic mesoderm. So mesoderm is, you remember, is an embryonic kind of tissue which will give rise to, for example, connective tissue or bones. So mesoderm is a term that we already know, but usually when we just say mesoderm, we mean the mesoderm that will actually form the body of the future baby. But here we also have mesoderm which is not going to be part of the embryo, so it's extra embryonic mesoderm. So it will be part of this red area here. And outer side, meaning pointing towards the maternal part, will be so mesoderm, you can imagine it as like loose connective tissue. So it's some kind of tissue like this, but it has cells that can still differentiate. So extra embryonic mesoderm, and then you have cytotrophoblast and syncytiotrophoblast, which will be part of the chorion as well. And we will see these two in the placenta, in the villi of the placenta. So the chorion will become part of the fetal placenta. So the maternal placenta will develop from decidua basalis. So decidua basalis, this orange area, will become maternal part of placenta, and the fetal part of placenta will be this, the chorion. So the chorion meaning both the extra embryonic mesoderm, so something like a connective tissue, and then cytotrophoblast and syncytiotrophoblast, which are going to be composed of cells, not so much extracellular matrix like in connective tissue here in the mesoderm, but these will be two types of cellular tissue that we will see in placenta and in the fetal membranes as well. And the cells of the chorion are important because as they proliferate, they divide, they start producing a lot of hormone called human chorion gonadotropin. So human H choriogonadotropin. So this hormone is the hormone that we also kind of mentioned last time, because it's the hormone that will inhibit the degeneration of the yellow body, the corpus luteum. When the egg gets fertilized, all of this happens and the chorion starts proliferating. It starts producing the HCG, which is as well the hormone that is often measured in the pregnancy tests. So this is the HCG that the over-the-counter pregnancy tests are using for detection usually. So it's inhibiting the degeneration of the corpus luteum, and therefore the gestation can continue. And the same way as we divided the decidua, so that we had decidua basalis, parietalis, and capsularis, the chorion is also divided based on where it's located, relatively to the embryo. So at the place where the blastocyst originally nidated, where it made the nest inside the wall of the uterus, so at this area, which is also called the embryonic pole, so it's the place where you have the embryo inside the uterus. It starts to grow, so then it will fill the whole uterus, but at the beginning it was here. So at this area the chorion will form a lot of villi, and this is the area, once again, like the part of decidua basalis, which will become the placenta. So here in this picture you already see this is decidua basalis and this is the so-called chorion frondosum. So a lot of Latin terms, I'm afraid, but chorion frondosum is this part which has a lot of villi and will become part of the placenta. the rest of the chorion, which goes around the embryo underneath the decidua capsularis, is called chorion leve. So it's basically just the part that's not going to become placenta, and it doesn't have villi. It doesn't have so many villi. And it's not going to become part of the placenta. So chorion leve is all of this red, I clicked, all of the red area, or basically almost black here, black line here, and chorion frondosum is this part, fetal part of the placenta, because chorion is part of the embryo, like it developed from the blastocyst, so it's the fetal part, it's not maternal tissue, and this part will be maternal. This is the decidua basalis and decidua parietalis and capsularis. So this was chorion. Yes, it's specifically the syncytio trophoblast part of the chorion that will secrete the human chorionic gonadotropin hormone. Perfect. So this was chorion, and so I told you that there are some villi that the chorion is making in the area where it will become fetal part of the placenta. And so the villi you can imagine similarly to what, for example, we saw in the small intestine. So like appendages coming from the wall, which are just freely like floating in the surrounding space. And depending on their development, the chorionic villi can be divided into three sort of groups. And you also have this in your workbook. So let me just go there. Let's skip what we will see in a while. And here you have one point in the workbook where they tell you to draw cross-section and cross-section of primary chorionic villus, secondary chorionic villus, and tertiary chorionic villus. So that's what we will be looking at right now. And you should specify the barrier between maternal and fetal blood, so we will get to that. But the three stages of development of these villi will be first the primary villus, secondary, and then tertiary, which is the most developed one. So primary villus will just be cytotrophoblast and syncytiotrophoblast. So it's chorionic villus, so it's just chorion. And the chorion will, at the beginning, it will be just cytotrophoblast and syncytiotrophoblast. So to remember which one is which, syncytio means that more of the cells are fusing. So the cells will have more than just one nucleus and they will be fused. So we will take a look at that under the microscope. In cytotrophoblast, it will be one cell, one nucleus. So this is a primary villus. It doesn't have like an inner sort of loose area that we will see later on. When the development continues, the villus is now being called secondary, and it starts to have a core which is filled with the extra embryonic mesoderm. So when I introduced the chorion, I talked about extra embryonic mesoderm on the inside and cytotrophoblast and syncytiotrophoblast on the outside. So here we are. So we have extra embryonic mesoderm, so sort of like loose connective tissue looking tissue. And surrounding it, we will have cytotrophoblast, so a layer of one cell, one nucleus, and then the syncytiotrophoblast, which is on the outside, and it's fused cells, a lot of them. So you will see cells which will be big and have more than just one nucleus. So this is secondary chorionic villus, and how to differentiate it from the tertiary chorionic villus is that here you will start seeing blood vessels inside. So you still have some cytotrophoblast, but it will start to disappear. So it will not be there like always. It will start disappearing. You will still have the syncytiotrophoblast, the darker green one with the more nuclei in the cells. You will still have extra embryonic mesoderm, so the mesoderm that is not going to become part of the embryo, and inside it you start seeing capillaries. And these capillaries will contain the blood of the fetus. So because the chorion is part of the fetal part of the placenta, so it's originally part of the fertilized egg. It came out of that. So it's not maternal. Therefore, the blood vessels that are inside the villi will contain blood of the embryo. This will be important later on. So we have one more picture for you to imagine this, and we will take a look at it on the placenta slide as well. So primary chorionic villi are coming like this. Secondary will have extra embryonic mesoderm in the middle. Tertiary will have blood vessels of the embryo. So this is the fetal side, so the blood supply comes from the fetal side, and this is the maternal part, which will be the decidua basalis in case of placenta. Yes. Okay, perfect. So hope you didn't quit the video yet. So let's carry on. So this was chorion, and now what about amnion? So amnion is still here, and it's the inner fetal membrane. So amnion is the inner one, chorion is around it, allantois we will get to is somewhere in between. And so amnion is lining the amniotic cavity, and that's the cavity in which the embryo is floating. So there is some hollow space, but this is not the amniotic cavity. This is just the cavity of the uterus, which will, in the end, not be there, because the amniotic cavity and the embryo will get larger and larger, and they will fill the whole space. But originally, the amniotic cavity is just here around the embryo. So you have amnion, and then you have chorion, and then you have decidua capsularis. And it also will cover the umbilical cord. So the amnion, which is lining this cavity, will also line, of course, it makes sense, it will line the umbilical cord, which is going from the placenta to the fetus. So all of this whole cavity will be lined with amnion. And at the end of the 3rd month of the gestation period, it will, because the fetus will grow so much that the amnion here, this thin black line, will get close to the chorion and it will fuse with it. So you can imagine this tiny embryo and the amniotic cavity to getting larger and larger until we hit the chorion. The chorion, which is chorion levae, which is this part. Chorion frondosum is this part, the future placenta. So we hit the chorion with the amnion going closer and closer because the embryo is getting larger and larger. So these two will become one layer, basically. So here you already see that the chorion levae and amnion are fused. So the chorionic cavity, which was this one between amnion and chorion, has already disappeared. So this has increased in size, this part, the amnion with the embryo, and it reached the place where you have the chorion, so this will become the chorioamnion or amniochorion. So these two layers will fuse, and you can see them here in black. So it's chorion leve, this area of chorion, and amnion. So this is the first thing that will happen, and then the embryo and the amniotic cavity expand even more, and then this whole thing will grow all the way to the wall, the free wall of the uterus. And what is going to happen now is that this part of the endometrium that changed into decidua capsularis, this area of endometrium on the surface of this whole embryonic stuff. So this surface will get all the way close to the wall of the uterus. So the uterine cavity will not be here anymore. So we already filled the chorionic cavity with amnion. We got all the way close to chorion and it fused. So we have amniochorion, and we still have decidua capsularis surrounding the amniochorion. And it grows, the embryo with the amniotic cavity and everything, until it reaches these sides of the uterus, where we have, because we already talked about this, the decidua parietalis. And at this point, the decidua capsularis, so this one, and decidua parietalis will fuse as well. So we will not have this huge uterine cavity, it will just be filled with the amniotic cavity with the fetus inside. And if the gestation progresses and it grows even more, the fetus, it starts to fuse the amniochorion with the two deciduas together. So in the end, all of these fused layers is what we call the fetal membrane. So here we have the layers separated. So we have amnion, then we have a cavity, then we have chorion, then we have one decidua cavity, second decidua. But when all of this results in a huge embryo inside a very big amniotic cavity, no cavity of the uterus almost, it will all fuse, all of these layers, and it will be the fetal membrane. And it's fused, decidua parietalis and decidua capsularis and chorion leve and amnion. And we will take a look at this as well. So we will have the opportunity to see all of these layers. So the first question is the chorion frondosum and chorion leve part of the chorionic cavity? So the chorionic cavity is the space between chorion leve and chorion frondosum and amnion. So they are forming the wall of the chorionic cavity. I think that's understandable now, hopefully. How does the secondary yolk sac disappear? So we are getting to the yolk sac, so hang on a little while we are already approaching the yolk sac. So here we are. So the yolk sac is another structure that's a temporary one, meaning it will disappear during the whole process of formation of the embryo. So part of it will be used for the development of intestine, but otherwise it will just degenerate and disappear. So at the 3-week-old embryo that we can see here, so we have amnion and allantois, which we haven't mentioned yet, and we have the yolk sac here. So if I let's get back, did we have a nice picture for this. Yes. So here at the beginning you have the chorionic cavity. So this will be the chorionic cavity that we saw on the images following that. And here you have the amniotic cavity, but you also have something we haven't discussed yet, and that's the yolk sac. So the yolk sac is here close to the embryoblast, and we will now talk about what happens to it, basically. So primitive yolk sac is what it's called at the beginning, and then secondary yolk sac when we are already in this stage with the big chorionic cavity. So let's go back to yolk sac here. So this is where we are, and like quite quickly this will change from one big sac to this kind of invaginated structure, which will become part of the embryo. So this is the blue area is the part of the embryo, and it's bending in a way, and the yolk sac part of it is becoming the future GI tract, and the rest of it is remaining outside of the future fetus. And at 5 weeks old, you also still see part of it that will be used for formation of the GI tract, and this part which was going outside of the embryo will be still there, but it will be inside the umbilical cord, and afterwards it will degenerate even from that. So it will just remain as like developmental stage of some of the parts of the GI tract, and we can still see it here going into the umbilical cord. Another important role of the yolk sac is that in this part of the embryo, of the whole embryonic tissue, this is the place where the future cells for the reproductive organs will start developing, and also it will become the place for red blood cells or like all blood cells to be produced. So red blood cells will start to be made, and they will then circulate in the capillaries inside the chorionic villi. So you will see the developing not mature erythrocytes as well in there. So this is the yolk sac, and then we have a part of the yolk sac. So it's so this is the part where the embryo's head is, and this is the caudal part. So cauda means the tail. So this is the tail part of the embryo, and at this part you see sort of invagination or duplication of the yolk sac going into here, also into the future umbilical cord. And this is also one of the structures that will in the future have a role in forming some organ, and in this case it will become part of the urinary bladder and one of the ligaments of the umbilical cord. So we can see part of the allantois in the slide of the umbilical cord as well. So we will take a look at that. So this was the beginning of formation of the umbilical cord, really. So imagine that we have, let me check the questions. Yes, yes. So you answer your own questions, which is perfect. So the blue and yellow cell layer is the embryonic disc. I think that was related to this picture or this picture here. Yes, this will become the embryo, and all of the rest will become either the fetal membranes or the placenta. So yes. Yeah, how does the secondary yolk sac disappear? It like degenerates. So at some point it just disappears. So I'm not really I'm not really sure how to describe this process. Take a look at the presentations from the lectures. It will have more detail. So so now to the umbilical cord that we will take a look at in a while. So it's connecting the embryo to the maternal organism via placenta. So it's connecting the placenta and the fetus. It's like there are some like diameter and length, some information about that. And when we take a look at the cross section of the umbilical cord, we will see that it's covered with the amnion. So amniotic cells, in this case amnioblasts. So it's same as fibroblasts, it's just a cell of the amnion, and it will be lining the whole umbilical cord. So this blue layer could be it. Then it will be filled with a special kind of connective tissue that we have already seen in the practice, and it's Wharton's jelly, so we will not deal with that too much. And it has a blood supply for the embryo, which will be 2 arteries and 1 vein. And sometimes you will have more of the blood vessels on the section of the umbilical cord. That's because the umbilical arteries sort of wrap around and wriggle around the umbilical vein. So you can have more sections than just, 1 artery and 1 vein and 2 arteries on one cross section. So this is the umbilical cord, and then we will also take a look at the placenta. So in case of placenta, we already got to the point where we know that the fetal part of placenta will be the chorion frondosum with the chorionic villi, and maternal part will be decidua basalis, the changed endometrium. And between these two, there will be the so-called intervillous space. So this is the umbilical cord, and this outer side will be the maternal tissue. So this will be close, this will be like changed part of endometrium, and this part will be the fetal part, the part where you see the chorionic villi. And this space between the villi is the intervillous space, and it's lined with syncytiotrophoblast, which is covering the villi. So we already saw the cross sections of the different stages of villi, and it had syncytiotrophoblast part of the chorion on the surface. And some of the villi will be called so-called anchoring villi, and these will be the ones that are attached to attached to the maternal part. So they will reach all the way from this part of the fetal placenta to the maternal part, and they will sort of connect to it, so they will sort of anchor themselves to the other side of placenta. And the rest of them that do not connect to the other side, to the maternal tissue, are called free villi. So they are just dangling in the intervillous space. And here's where we come to the placental barrier between the two types of blood that we have here. So we have the maternal blood, which is of course inside the maternal tissue. And then we have the newly formed blood vessels of the embryonic tissue, where you have the blood of the embryo. And we need to separate these two from each other because, for example, to some point, the embryo is a foreign organism and it has different antigens and stuff like that on its cells. So the contact between the maternal immune system and the fetal blood wouldn't be, the direct contact wouldn't be very good. So we want to have some kind of barrier between these two, and that's basically the wall of the villi, because maternal blood will be everywhere here in the intervillous spaces, and then you have the wall of the villus, and inside in the extraembryonal mesoderm you have the blood vessels of the embryo. So for the gas and metabolite exchange, the maternal blood, which is oxygenated, comes here and it has nutrients. It comes here to the intervillous space of the placenta, and then the metabolites and the gas has to cross the wall of the villi and get into the bloodstream of the embryo, and like vice versa. So when the embryo has already used the oxygen and the metabolites, the blood comes via the blood supply back to the placenta from the fetus, and the gases and metabolites will cross this barrier once again. So let me check if we have any questions. So the yolk sac enters the connective stalk. Yes, that's right. That's right. Are intervillous space and lacunae the same thing? Yes. Yes, it's just a different word. So don't worry about it. It's the same thing. We use this one, so probably use this one as well. But in the textbooks you can find all sorts of different things. So this is a video on the development of placenta. So it's once again like a 3D video for you to imagine this whole process. So I will play it and you can get back to it if you want to. So it's almost 4 minutes, but we might skip
Development
some
of the placenta begins during implantation. When the blastocyst adheres to the endometrial epithelium, trophoblast cells differentiate into an inner cytotrophoblast layer and an outer syncytiotrophoblast layer. As the cytotrophoblast proliferates, newly formed cells migrate into the syncytiotrophoblast and lose their cell membranes, forming a rapidly growing, multinucleated mass. The cytotrophoblast secretes proteolytic enzymes, and the syncytiotrophoblast sends out finger-like projections, allowing the blastocyst to embed into the endometrium. Lacunae, or spaces, begin to form within the syncytiotrophoblast. As the syncytiotrophoblast erodes the endometrial blood vessels and glands, the lacunae become filled with maternal blood and glandular secretion.
Exactly. So here
Isolated lacunae fuse to form a network through which maternal blood flows, thus establishing early uteroplacental circulation. Toward the end of the second week, small projections of the cytotrophoblast begin to expand into the syncytiotrophoblast, forming primary chorionic villi. Early in the third week, extraembryonic mesoderm grows into these villi, forming a core of loose connective tissue, at which point the structure is known as secondary chorionic villi. By the end of the third week, embryonic blood vessels have begun to form in the extraembryonic mesoderm of the secondary chorionic villi, transforming them into tertiary chorionic villi. Cytotrophoblast cells from the tertiary villi grow toward the decidua basalis and spread across it to form a cytotrophoblastic shell. Villi that are connected to the decidua basalis through the cytotrophoblastic shell are called anchoring villi. Villi growing from the sides of the stem villi are called branch villi. These branch villi are surrounded by the intervillous space and will serve as the main sites of exchange between mother and fetus. By the fourth week, fetal blood flow is established. Poorly oxygenated blood is transported to the placenta from the fetus via paired umbilical arteries. These arteries send branches into the chorionic villi and divide into capillary networks at the terminal ends. Carbon dioxide and waste products are removed from the fetal circulation across the placental membrane to the maternal blood in the intervillous space. Oxygen and nutrients are transported across the placental membrane from the maternal blood to the fetal capillaries. Oxygenated fetal blood travels back through a vein, converging to form a single umbilical vein that goes to the fetus. Blood returns to the maternal circulation through endometrial veins, forced out by the pressure of incoming arterial blood. The placenta nourishes the fetus throughout development and is delivered shortly after parturition.
So this was just a repetition of what we went through, and this was the formation of the placenta, quite nicely visualized in 3D. So maybe that will help some of you imagine how it goes. Yeah, exactly. So, Lacune, here we go. What is the difference between the splanchnic, somatic mesoderm with chorion frondosum and chorion leve? Is it different names for the same structure, mesoderm wall? What is the So the mesoderm, for example, the somatic mesoderm, it will give rise to structures that will be part of the embryo of the future baby, whatever. So that's the mesoderm. That's part of the fetal area of the whole structure, and the part of the chorion is also mesoderm, but it's the extra-embryonal mesoderm. So I hope this is the answer to this question. Hopefully. If not, we will get back to it. So this was the placenta in 3D. This is just another image for you to visualize everything that we have talked about. So you see the maternal part of the placenta, so decidua basalis, and then you have the maternal blood vessels going into releasing the blood and taking the blood out of the intervillous space, and then you have the chorionic villi, which grow from this side, from the embryonic pole of the placenta, and they become tertiary when the fetal blood supply blood vessels develop. And so between the intervillous space and these blood vessels is where you have the placental barrier, which is important, for example, for stopping some hormones to go through and making some others to go through the placenta towards the fetus specifically. And this is what it will look like under the microscope. So we will take a look at this in a while. Maternal side will have the decidual cells that we have already seen. Fetal side will be the chorionic plate with the villi coming out of it, and some of them reaching the maternal side, and those will be called the anchoring villi. And here we have one slide concentrating on the barrier again. So you have the list of the structures that are forming this barrier. So you have the maternal blood in this area, inside the intervillous space, and this would be a cross section of one villus and another one here and here. And so first of all, if for example, the blood components and carbon dioxide needs to go from the fetal blood to the maternal part, it will first need to cross the endothelium of the fetal blood vessel. So typically a capillary. So endothelium is the first layer that we have to cross. And then, of course, the basement membrane of the fetal capillary endothelium. Then all of this loose tissue is the extra embryonic mesoderm. So extra embryonic mesoderm or mesenchyme is this loose tissue. So it needs to go through this loose tissue, and then it reaches the wall of the villus. And the wall of the villus is composed of cytotrophoblast. So cytotrophoblast is this layer, and then the basement membrane of the cytotrophoblast, of course, and then the syncytiotrophoblast. So that's the outer layer where you have fused cells, like big mass of fused cells, like you saw it in the video, and then it can reach the maternal blood, which washes all the villi in the intervillous spaces. So we have come to this stage where we have the placenta from chorionic villi and the chorionic plate, and the blood supply of the embryo, the blood vessels come via umbilical cord into the placenta, into the villi, and the exchange of gases and nutrients happens across the wall of the villi between the blood vessels of the embryo and the intervillous space where you have the maternal blood. And now we will continue from the theory, we will go to the slides. So I will once again check the chat. When we talk about chorionic plate, from which sublayers is it formed? Okay, so yes, it's something I haven't really mentioned before. So the chorionic plate will be composed of the extra embryonic mesoderm, so the same stuff that's filling the villi. So I will make it larger. So the chorionic plate is this area, and the villi come from it. So it's filled with the same stuff that the villi are filled with, so that's extra embryonic mesoderm. It has blood vessels in it that are going from the umbilical cord and to the umbilical cord, and it is so on the side facing the intervillous space, it will be lined with the trophoblast. So both syncytiotrophoblast and cytotrophoblast. So the lining of the villi and of the plate is the same. Then you have the mesoderm, and then on the other side we are facing the amniotic cavity. So here we will have the amnionic epithelium. So amnion, thin layer here, which fused with the chorion here, as well as in all the other areas of the wall of the uterus. And so extra embryonic mesoderm and lining with one side from amnion, on the other side from trophoblast. So I think that's it. We will take a look under the microscope as well. Perfect. So now let's continue with the slides. So the first one is very easy. It's the umbilical cord that you have already seen before. And today we want to not just focus on Wharton's jelly, which we have already seen, but we want to as well focus on the 2 arteries and 1 vein, and we want to take a look at the amniotic epithelium, which will be lining the umbilical cord from the outside. So the lining, then the Wharton's jelly, the structures that we see here, which will be 2 arteries, So in this case, number one are arteries, and number 2 is a vein. And the arteries, they have thicker wall and they are more rounded. They have more smooth muscle in the wall, and the vein will be more collapsed with a thinner wall in general. So this is what it will look like from a close-up. So this would be a section of the wall of one of the arteries. You can see here that we are aiming for the eye of the smiley face, not so smiley in this case. So all of this is smooth muscle, some connective tissue, and this would be the lumen of the umbilical artery, lined with a simple squamous endothelium, some red blood cells hanging in here. So this is the wall of the artery, and everything that you see around here will be the Wharton's jelly. So it's much looser and it contains spindle-shaped cells, which are the fibroblasts. So a close-up here, and you see the surface as well. We will go through the online virtual slide as well, but this is quite a nice picture from the PDF that I told you that's in Vyuka, the slides PDF. And so this is the amniotic epithelium, the amnioblasts, so the cells of amnion that are lining everything inside the amniotic cavity, which is the umbilical cord as well. So amniotic epithelium and Wharton's jelly everywhere here. This is the Azan stain, which is in the slide box that we have for the practices usually, and so it's kind of purple. But it could be more pink as well, and fibroblasts, so the cells that are forming all of these extracellular matrix of the Wharton's jelly are fibroblasts sitting here. And so the Wharton's jelly is a jelly, so it has a high amount of glycoproteins because they bind a lot of water, which makes the final sort of consistency quite jelly-like. So you would also have them in cartilage, for example. So this is the umbilical cord, and in the workbook you have that you should be able to identify what's the Wharton's jelly. So that's very easy. That's like everything that's filling the umbilical cord, and you should be able to differentiate the umbilical arteries and the umbilical vein, and you should be able to find the simple cuboidal epithelium of amnioblasts lining the umbilical cord. So if we go to the chat as well. Yes, okay, perfect. No more questions right now. So if we go to histology guide, there is a slide of umbilical cord and its hematoxylin eosine. So we can take a look at the whole structure. So this is a cross section, and it's a nice one. So it has really just 3 vessels, not more sections of those. And you even can, let them help you with the differentiation between the arteries and the vein, which is in this case quite useful because it's not very clear. So usually it's clear. So these 2 will be the umbilical arteries, and this one will be the vein. But in this case, it doesn't look much thinner, and it's not, collapsed at all. So in this case, it would be hard to distinguish the arteries and the veins. So I think it wouldn't be required of you if it was this complicated. So these are the arteries and the vein. If you zoom in to the wall, you should be able to identify that this is smooth muscle, a lot of smooth muscle, and here we can really nicely see the endothelium lining the lumen of the artery. So smooth muscle, And as we move out here, you can clearly see this is the border where we start to have the Wharton's jelly. So it's very loose, and you see nice spindle-shaped cells with elongated hematoxylin state nuclei. So these are the fibroblasts, and everything else is just extracellular matrix. So extracellular matrix of Wharton's jelly and the fibroblasts. And then we want to take a look at the surface as well. And here we see the amnioblasts. So it's more or less a simple cuboidal epithelium. So you see that the cell nuclei are round. So it's a cuboidal epithelium. Sometimes there's like more layers, so it doesn't really matter. But it's the amniotic epithelium, amnioblasts, lining the surface of the umbilical cord. So that's all that you should be able to find on the slide of umbilical cord cross section. And there is one more structure here, which is the allantoic duct, but it's not necessary for you to know anything about it right now. So you should be able to identify the blood vessels, to find Wharton's jelly, which is very easy, and to see the amniotic epithelium. So I think for umbilical cord that should be enough. So let's continue. And so the second one will be more complicated. So let's take a look at the placenta. So first of all, there are differences between placenta at the beginning of the gestation, at the beginning of pregnancy, and in the final period of the pregnancy. So, but in general, what we will see in placenta will be the maternal part, so the decidua basalis, so decidual cells, we will take a look at them, and then the fetal part, which will be the chorionic plate and the villi coming out of it, and it will be filled with the extraembryonic mesoderm, and between the villi we will see maternal blood, and on the surface facing the amniotic cavity, so the surface coming further away from the maternal tissue of the chorionic plate, we will see the amniotic epithelium. So the same one as we saw in the umbilical cord. So we are still in the same cavity. So part of the placenta and the umbilical cord, everything will be covered with the amnion. So this is once again the picture that I have in the presentation. So this is an overview. So if you had the whole round structure of the placenta, the disc, you would cut across. So you would cut across the disc, and you would have the maternal side, the decidua basalis, fetal side, the chorionic plate with the fetal blood vessels, and the villi branching from the fetal side, and between them the intervillous space. This is a close-up a little bit, so we start seeing the decidual cells here, but we will move into a higher magnification, and here you see the chorionic villi. So the placenta is very different from all the other slides that we have seen, because it's a lot of empty space, and inside it these cloudy structures with quite loose inside usually, so quite light. And we will see the difference between the villi that we see at the beginning of the pregnancy and at the end. So there will be villi all the time, but based on their composition, how they look, we will be able to say if this is first trimester or the third one. And at the first trimester stage, the the villi, like they are already the tertial villi. They already have the blood vessels of the fetal blood supply inside of them, but they still have both cytotrophoblast and syncytiotrophoblast. Inside, it's extraembryonic mesoderm with capillaries, blood vessels. And as the pregnancy progresses, the villi change and we stop seeing cytotrophoblast almost like completely. It will be mostly just syncytiotrophoblast covering the surface of the villi, and inside we will have more fetal capillaries and more bigger ones. The size will change and the number will change. So in general, the differences are that in the third trimester there will no longer be the cytotrophoblast visible in the wall of the villus when you have it on cross section, and the other difference is based on the red blood cells that we will see inside the blood vessels inside the villi. So during first trimester there are still not mature erythrocytes. There are erythroblasts which still have nuclei. So this could be, or this is, a picture of a villus from the first trimester. So this is the extraembryonic mesoderm, the loose tissue inside the villus. This is the intervillus space, and this is the wall of the villus. So you see that we have cytotrophoblast, one layer of cells, and syncytiotrophoblast, another layer of fused cells. And so that's one thing that's telling us it's probably first trimester, because we have both of these, cytotrophoblast and syncytiotrophoblast. And another thing that can help us to differentiate is if we find a capillary like this, a blood vessel inside the extraembryonic mesoderm, and inside you can see these cells will in the future become red blood cells, and they are the erythroblasts, which still have nuclei. And when it's the third trimester placenta, you will already see normal red blood cells that we are used to in all the other slides that are, from adults. So this is the overview of the differences, how to tell between first trimester placenta and third trimester placenta. No more questions? You're probably asleep now. No. So we have some pictures now from our slides because the histology guide, it's not embryology guide so much, so it just has one picture of placenta, and it's so it doesn't have the comparison between the two types. So we will take a look at that one as well, but first of all, let's see all of the structures on the pictures in the presentation. So this area, so you would have a slide, tiny magnification like this, so it's a lot of mass, and it usually has two sides, and on one side you will find the chorionic plate, and on the other side you will find the maternal part, so something like this. So the fetal part, the chorionic plate, a lot of villi in the middle, and the maternal part, and our job is to be able to say which part is which, basically, and to find them. So in this case, this would be the chorionic plate, and this would be the amniotic amnioblasts, the amniotic epithelium lining the surface facing the amniotic cavity. So this is a lot of chorion here and a lot of extraembryonic mesoderm with some blood vessels of the embryo. So this is the embryonic part of placenta. We will go into close-up, and these are the villi that are coming out of the chorionic plate. And the white space is the intervillous space. On the other side, we will see again like a pink area, which is not filled with villi, so it's solid pink. And when we zoom in, we will see the decidual cells. So we will find out that it's the maternal part. To just first take a look at the villi. So this is a close-up of the chorionic villi, and you see that you have a lot of blood vessels inside of them. So extraembryonic mesoderm filling the villi with blood vessels filled with red blood cells, and the lining of the villi here, it's just one layer. It's not like cytotrophoblast and syncytiotrophoblast. This is just syncytiotrophoblast. So this would be the case of third trimester placenta. So the red blood cells, they don't have nuclei. They are the usual ones, the normal ones, biconcave shape, red blood cells. And so they are the fetal blood cells, red blood cells, and in the intervillous space you have the maternal blood. So maternal red blood cells will always be the mature ones, of course. And we only have one layer covering the villi on the surface, and that's the syncytiotrophoblast. So this would be the third trimester. And now let's see the first trimester placenta. So this is like just the villi now, and we will focus on the decidua and the chorionic plate in a little while. So this is like a small magnification again, and so these will be the villi filled with the mesoderm, extraembryonic mesoderm again, and the lining of them, and some maternal blood here in the space between the villi. And this is a small close-up, and we will go to an even larger close-up. So this is the intervillous space, and this is one villus with the inside of it filled with extraembryonic mesoderm, and here we have a blood vessel. And this is the wall of the villus, and here you see that this is the syncytiotrophoblast, so the fused cells that we have lining the like facing the intervillous space, but we also have one layer of these round like sort of cuboid cells, and this is the cytotrophoblast. So we have one layer, second layer. So this is telling us it's probably first trimester. We want to check, so we take a look inside the blood vessel, and we see erythroblasts still with nuclei. So this is clearly first trimester placenta. Another thing that I did not mention, so we also have, oh yeah, I have it it's coming in 2 slides. Sorry, I didn't forget about it. There are also special macrophages located in the chorionic villi, and they have a special name. Like you have Kupffer cells in the liver, so you have Hofbauer cells in the chorionic villi, and they are macrophages located in the like extraembryonal mesoderm, in the connective tissue inside the villus. So they would be large round cells like this sitting in the extraembryonal mesoderm. They are there in their highest numbers at the beginning of the pregnancy. Their function is just, cellular, non-specific immunity. So if we go back to this picture, you can see here an example, and second one, example of the Hofbauer cells, the macrophages sitting in the extraembryonal mesoderm. So this is first trimester placenta, and this is second trimester. So once again, to compare, you have a lot of blood vessels, and they are all filled, the fetal blood vessels, they are all filled with mature erythrocytes, and you don't have the cytotrophoblast anymore. You just have the syncytiotrophoblast lining the villi, facing the intervillous space. So do we have any questions? So the fetal red blood cells still contain nuclei? Yes. Yes, you're right. At the beginning of the pregnancy they do. And then they mature, they become the normal mature erythrocytes during the prenatal period. But in the first trimester, they still have nuclei. So they are like the precursors of the red blood cells. So they are not fully formed yet. So in the workbook for placenta, we have staining hematoxylin eosin, okay. And yes, so we should be able to indicate the stage-related structural features. So you should know basically the difference between first and third trimester. So that's the absence of cytotrophoblast in the third trimester, and the changes in the red blood cells. So in the first trimester, you would still have the nucleated erythroblasts. And so we should be able, so now we will go to histology guide and take a look at the whole placenta slide, and we should be able to find the chorionic plate and the villi coming from it. So this will be the fetal part of placenta. And then we should, on the villi, we should be able to see the syncytiotrophoblast and cytotrophoblast. If it's the first trimester, if it's the third, we will only see the syncytiotrophoblast. We should be able to find the extraembryonic mesoderm, which will be inside the villi, and we should see the fetal blood vessels and their endothelium, which is part of the barrier between the blood of the fetus and the blood of the mother. And then we should see the maternal part of the placenta, the decidua basalis. And then you have the different stages of the villi which you have in the presentation. So let's start with the placenta slide. So here we go. So overview, it's one big complicated messy structure, but you see here is this big solid area, so we want to take a look at that. And on the other side you have something similar, sort of like a solid tissue, not so much, not just the villi. And all of this will be the villi of the chorion and the intervillous space. So let's first zoom in onto this side, this area. So we can now see that inside this solid tissue on this side of the placenta, we see very big, sort of swollen cells with light cytoplasm and big round nuclei. And so this should already remind you of what we covered at the beginning of the practice. So this is a beautiful example of decidual cells. So this is part of the maternal part of the placenta. This is the decidua basalis. So this used to be part of endometrium. It underwent decidualization, and now it stores a lot of glycogen and lipids for the nutrition of the fetus. So the change of the cells is very, very apparent here. So these are the decidual cells, and all of this is the decidua basalis. So we can be sure that this is the maternal part of placenta. So on the other side from here, we should see the fetal part, which is the chorionic plate with the amniotic epithelium, hopefully, as well. And all the villi that we see here are the fetal part as well. And they all come from the chorionic plate. They are all part of chorion. And the decidua basalis has blood vessels, and these blood vessels release the blood into the intervillous spaces. So maternal blood will be between the villi. So I hope the decidual cells are quite clear. They should be really easily recognizable, and when we go to the other side, you will see that there's nothing similar over there. So when we zoom in here, this is basically connective tissue. So you don't have to worry about mistaking these two. I don't think so. So this is the chorionic plate. So it's extraembryonic mesoderm, and it's lined with the syncytiotrophoblast, similarly to the villi. So the villi are branching off from this side of the placenta, and it's extraembryonic mesoderm, and the blood vessels it will contain will be the blood vessels of the embryo. So for example, here you see some blood vessels. So these are the blood vessels of the embryo, and all of the villi will be containing the vessels as well. So this is the chorionic plate. I will try to find if there's the amnion as well, but I don't think so. It's probably torn, which is quite easy to do. Yeah, but on this side of the chorionic plate, you would see a similar epithelium as here in the umbilical cord. So amnioblasts forming like cuboidal epithelium like this. So something like this should be ideally located on this side of the chorionic plate, and this would be facing the amniotic cavity. So chorionic plate, and now let's zoom into the part with all the villi, and let's try to see if this is a placenta from the first trimester or if it's from the third trimester. So let's see if there are any questions. Yes, no, welcome. And so let's zoom in, and so you can zoom in anywhere basically. It's the same everywhere. So if we zoom in, so there are the villi and the intervillous space. So we have blood vessels here and blood vessels here, and this is the extraembryonic mesoderm filling the villi, and then you have the blood vessels, and you could also have the Hofbauer cells inside the extraembryonic mesoderm. I wouldn't really want to say which cell specifically could it be. Not really. It's quite messy here, so we don't have to deal with that, I think. So how to differentiate between first trimester, third trimester? We want to look at the wall, the lining of the villi. We want to look at the red blood cells. So the lining of the villi is just one layer. So we don't see like cytotrophoblast and syncytiotrophoblast. We just see one layer, and that's only syncytiotrophoblast. You can never have just cytotrophoblast, so you don't have to worry about this. And this is layer of fused cells that's lining the villus. So this would point us into the direction of this being like third trimester, like late term placenta. And then we need to check the red blood cells, and here we see beautiful biconcave, ready to use erythrocytes. So this is for sure a placenta not from the first trimester but from later on. So just to compare again, with this example of the first trimester, you would have cytotrophoblast and then syncytiotrophoblast, and in the blood vessels you would have a lot of nucleated cells, which would be precursors of the erythrocytes. So this is I think everything that you need to see in the placenta. So we found decidua basalis. This is the maternal part of placenta, and it has the decidual cells, which are easy to recognize. And then we found the chorionic plate on the other side and the villi that are coming from it. And on the villi we found only syncytiotrophoblast, that's sign of third trimester placenta. Inside we had sort of loose tissue called extraembryonic mesoderm, and we saw the fetal blood vessels in which you can clearly see the endothelium lining here. So if we want to cross from maternal blood, if you want to move, for example, oxygen from here to here, we need to cross the syncytiotrophoblast and also the cytotrophoblast. If it's here, their basement membranes, we need to go across the extraembryonic mesoderm and across the basement membrane and the endothelium lining the capillary. And we are in fetal blood. So this is the placenta. So the chorionic plate is part of the chorion. That's just not forming villi. So if we go back in the presentation to the nice picture here, so you can see that there are the villi, but they come from this flat part of the chorion. So it's again extraembryonic mesoderm being lined with the syncytiotrophoblast and cytotrophoblast when it's pointing towards the intervillous space. And on the other side it will be covered with the amniotic epithelium, because we are in the amniotic cavity, same as umbilical cord. So we were looking at these villi, this chorionic plate, and the decidua basalis, the changed endometrium with the decidual cells. So I think that's hopefully the answer to your question. And let's move on to the last part. We have like 3 minutes, so it's probably be a little bit longer. I'm sorry. So finally, we will take a quick look at the fetal membranes. So that's the I will go back to the picture again. This is the best picture probably from today's presentation. So now we were looking at this area where we have the whole placenta formed, and the last slide will be the fetal membranes, which is like anywhere here if you had a cross section of all these fused layers. So we will have amnion fused with chorion levae, not chorion frondosum, that was part of the villi. So amnion, chorion levae fused into amniochorion or chorioamnion, doesn't matter. Then you have decidua capsularis and decidua basalis fused. So we will see all of these layers merged into one sort of wall, and it will be called the fetal membranes. So this is like the schematic overview of everything that we want to see in this slide. And so we can go, for example, from the embryonic side, so from the amniotic cavity. So we have the fetus in the cavity, and the cavity is lined with amniotic epithelium. So you have the same epithelium as was the one lining the umbilical cord that we saw, with a little bit of extracellular matrix underneath it. So this would be the amniotic epithelium, and then a little bit of connective tissue. So amnion is the first layer where we just really want to focus on the epithelium, and underneath we fused already with the chorion levae, so the chorion lining the uterine, the chorion that is not part of the placenta. And so chorion will be this area, and then we will have decidua, so the maternal part. So amnion and chorion, these two layers, are part of the fetal sort of tissues. So fetal part and maternal part, decidua. In this case, it's not decidua basalis, but fused decidua capsularis and parietalis. And both amnion and chorion will have like a layer of mesoderm to it. So we will have all of the mesoderm here. So mesoderm, extraembryonal mesoderm, or extraembryonic mesoderm. So this loose connective tissue again from amnion and from chorion. So together this will be extraembryonal mesoderm. So we'll have epithelium of amnion, extraembryonal mesoderm from both amnion and chorion, and then we will have the part of the chorion which is the trophoblast. So it will be cytotrophoblast and syncytiotrophoblast. So mesoderm is here between amnion and the cells of chorion, the trophoblast part, and the mesoderm is both part of chorion and amnion, but it's fused. So it moved so close together that it fused. So we have amniotic epithelium, extraembryonic mesoderm, cytotrophoblast and syncytiotrophoblast, and then the decidua, which will be once again very easy to recognize because of these huge decidual cells. So we have once again to review this part of the wall of the fetal membranes, and it's decidua, so the maternal part, decidua parietalis and capsularis, these two fused. It's chorion leve coming around the fetus like this, and it's amniotic epithelium, which is forming the amniotic cavity at the beginning, but now all of them fused together. And between amnion and the chorion cells, the cytotrophoblast and syncytiotrophoblast that we know from the villi already, there is the fused extraembryonic mesoderm of chorion and amnion. And this is what it will look like underneath the microscope. So there is no slide for fetal membranes that I have found on Histology Guide. So this is the one that we have pictures from the from our slides from the practices. So this is a very small magnification, but already you can see that it's like many layers, and it's a longitudinal structure like this. So this is the fetal membranes, and they would be going around the amniotic cavity. So we need to zoom in, obviously, and so the layers will be a bit torn away from each other, and there will be a lot of loose space. So the loose space that you will see here, the large loose space, will be the extraembryonic mesoderm. So that's the loose connective tissue, sort of, which tears and distends and is quite loose in general. So this side, so on one side of the fetal membranes, you will be able to find the amniotic epithelium. So that's how you identify which side is which. And on the other side, you will find the cells of the decidua. So decidual cells will tell you this is maternal part, the maternal side, and amniotic epithelium, so simple cuboidal, will tell you this is the embryonic part, the part which is facing the amniotic cavity. And so here we will zoom in even more, but here we will have the amniotic epithelium, then we will have all of this will be the extraembryonic mesoderm, so all of the connective tissue with the loose bits, and here as well will be the extraembryonic mesoderm of amnion and of chorion. And then you have so-called vascular layer, that's like part of the chorion already. It's not like terribly important. And here you will have the syncytiotrophoblast. So the trophoblast part of the chorion will be here, and attached to it, fused with it as well, will be the decidua. Capsularis and parietalis fused together, so you will not see the border very clearly here. So amnion, extraembryonic mesoderm, chorion, and the decidua. So this is the first part in large magnification. So number 1 is amnion. So you have the epithelium, and then you have the extraembryonal mesoderm. So you have amniotic epithelium with basement membrane, and then from here down you have the loose extraembryonic mesoderm. And the number 3 is just one cell of the epithelium. Number 4 is basement membrane. So it's a little bit of connective tissue, and then you have the extraembryonal mesoderm. And then when we move underneath the loose part, you see still extraembryonic mesoderm, and so it looks like normal connective tissue would. And then when you start seeing, many cells, so it stops being a lot of extracellular matrix, but it starts being all cells. It's the trophoblast and the trophoblast cells. So it's both syncytiotrophoblast and cytotrophoblast. You don't have to worry about it. It's the chorion part, which is cellular, composed of big cells, and it's the trophoblast cells. And when we go even deeper and we get to this side of the fetal membranes, you start seeing cells that will remind you of the decidua basalis that we saw in the placenta, and this will be the decidua, so the maternal part of the fetal membranes, and this would be one example of the decidual cell. So this would still be trophoblast, and this would already be decidua on the total bottom, on the other side from the amnion that we saw at the beginning. So let's return to the workbook and check if we have seen everything. So in fetal membrane slide, you should be able to find the amniotic epithelium. So when you zoom in to this structure under the microscope, you need to focus on the edges, and on one of them, on this one in this case, you will find a simple cuboidal epithelium. So this will be the side of amniotic cavity. So this will be amnion, and underneath it you would have extraembryonic mesoderm, then trophoblast cells, and then decidua, maternal part. So you need to find the epithelium, and then underneath it you will see the loose part of connective tissue, which will be both amniotic and chorionic mesoderm together fused. And then under this, if you go further down from the amniotic epithelium, you start seeing cells, one next to the other, big ones. So they will be part of the chorion, which is called the cytotrophoblast, and syncytiotrophoblast is just trophoblast should be enough. And then in the end, you see decidua, parietalis, and capsularis fused together. So that would be this area. So once again, on the top, the epithelium, extraembryonic mesoderm, trophoblast, syncytiotrophoblast and cytotrophoblast, and decidua, capsularis and parietalis fused. So epithelium should be easy to find, and underneath the loose part with the extraembryonic mesoderm you can find the trophoblast cells, and then on the total bottom these large cells with light cytoplasm that will be the cells of the decidua, the maternal part of the fetal membranes. So no more questions right now. So we went through everything concerning the extraembryonic organs. So thank you for your attention. And if you have any questions, because this is quite complicated and I'm not an expert, as you can see, so you can email me or some of my colleagues, and if you have any questions, concerning this or any other topic of histology. So this is it from today, and if you have any questions, you can continue to chat here. If not, just email me whenever, and we will see each other next time. No, I think I will not be the one doing the practice with the chick embryo. So you will receive information about the following steps and the test, hopefully soon. So thanks very much, and take care and have a nice day. Bye.